Hello everybody. My name is Louise Arno. I'm a PhD student at the KU Leuven, and today I will talk about this mean field theory for wave dynamics in the anisotropic cardiac wall. First, I will briefly introduce you to some mathematics of these cardiac arrhythmias, and then we will discuss this mean field theory for cardiac rotors. But the first question we should ask ourselves is, why does my heart beat? Well, these contractions are led by electrical signals that move through the cardiac wall. And these walls consist out of myocytes, which are cardiac muscle cells that are able to conduct and that are connected with each other. One of the most important aspects of these electrical signals moving through the myocyte is the transmembrane potential. And we denote this transmembrane potential with V, and is for every cell depending on time t. And over here we see one such cycle of the transmembrane potential. And we will use this variable to model cardiac arrhythmias. So how does this work? How do we go from the cell to the mathematical equations? And for this, we look at the reaction-diffusion equation that describes the evolution of the variable u, which consists out of this transmembrane potential V and the vector u. And this vector represents the ionic currents moving through the cardiac wall, but also the gates in the cell membranes that regulate these currents. Now, looking at these equations, we see that it consists out of two parts. Namely, the first part is the reaction part, which we see here in blue. And these are basically the same nonlinear ordinary differential equations that describe those chemical reactions or pandemics, we know. But the meaning of these blue terms makes them hard equations, in the sense that, for example, Cm is the capacitance of such a myocyte. When we look at the second part of the RD equations, which is here depicted in red, it makes the reaction-diffusion equation partial differential equations, and it tells us how this transmembrane potential moves in an anisotropic way through the cardiac wall. Now, how does this anisotropic diffusion of the potential V works? And for this, we have to look at the myocytes again, which over here are depicted in pink. And by looking at them, we see that they are stronger coupled in one direction, which we call the fiber direction, than they are in the perpendicular ones. And this stronger connection causes the transmembrane potential to move faster in the fiber direction than in the perpendicular ones. And this uneven spread of the transmembrane potential we call rotational anisotropy. And this is captured in the diffusion term of the reaction-diffusion equations. More specifically, it's captured in the diffusion tensor, which we denote by Dij. And this is a 3x3 matrix for which the eigenvalues in the direction I is proportional to the squared of the conduction velocity in that same direction. So, for example, when the speed in the fiber direction is 3 times bigger than the ones in the perpendicular, then the corresponding eigenvalue of the diffusion matrix will be 9 times bigger than the ones in the perpendicular direction. When we look at the curved space formalism, we can connect this diffusion matrix with the metric tensor, in the sense that the hard metric tensor is the inverse of the diffusion tensor. Now, we were able to connect these electrical signals moving through the cardiac wall with mathematics by using the reaction-diffusion equations. But now we would like to connect cardiac arrhythmias with mathematics. And for this, we will have to look again to this very nice picture. So we see over here 3 points of view, namely the ECG seen by the doctor, the simulation parts done by us basically, and the experimental point of view. And each point of view is divided into 3 parts, for which in the first part we see a normal heart rhythm, which is translated into a plane wave during simulations. In the second part, we see ventricular tachycardia, and this is a cardiac arrhythmia for which the lower part of the heart moves faster than a normal heartbeat. And this is translated into a 2-dimensional cardiac rotor during simulations, which is also confirmed by experiments. And in the third part, we see ventricular fibrillation, and this means that the lower part of the heart contracts in a very chaotic way and also very poorly. And this we can connect again with multiple spiral waves in our simulations. That is again confirmed by experiments. And so we see a clear connection between cardiac arrhythmias and cardiac rotors. But what are these cardiac rotors? Well, in our heart is a 3-dimensional object. So we need some kind of 3-dimensional cardiac rotor, and this we call a scroll wave. And these scroll waves are basically stackings of spiral waves. So when we take one slice of such a scroll wave, we get one spiral wave. And looking at these spiral waves, we see that they rotate around one point over here. And this point we call the tip of a spiral wave. And when we stack these tips on each other, we get the rotation axis of the scroll wave. And this is, again, the filament of the scroll wave. And for us, it's very important in our mean field theory. Now, when we do some simulations in a real heart geometry, we see that these scroll waves aren't perfect stackings of spiral waves. So these spiral waves are deformed or they are shifted with respect to each other. And this causes the filament to be bended or to be curved. So we don't have a perfect straight line of the filament, but the filament is curved, which we see over here. So I think it's best to think about this filament as a cosmic string, a tornado, or a hurricane. Now, as I said, these filaments are for us very important in the sense that they are also related to the complexity of cardiac arrhythmias, namely that the number of filaments classify the type of cardiac arrhythmias. Now, what do we try to do? We try to capture the dynamics of such filaments specifically for the cardiac wall. Now, how does this work? Well, there are already some theories that try to describe the dynamics of such filaments, but they assume that these filaments are very long and that they are very, very thin. So you can say that they are some kind of tornado theories. But there is a problem in the sense that for the ventricles, so the lower part of the heart, over there, that the diameter of such filaments are approximated by their height or by the thickness of the ventricles. And in the atria, that over there, the diameter of the filaments are even way bigger than their heights, so bigger than the thickness of the atria. So the question is, should we move from a tornado theory towards a hurricane theory, being our mean field theory? And for this, we already did some simulations where we compared the trajectory of a filament at a certain thickness of the heart wall with the tornado theory, so the classical theories here depicted in red, and our hurricane theory, which is our mean field theory. And by looking at these simulations, it seems that this hurricane theory seems to be a better fit. Now, how does this mean field theory works? Well, the calculations work in 2 steps. And in the first step, we introduce a new kind of system, a new kind of coordinate system, which we call the heat coordinates. And we denote these heat coordinates with Q1, Q2, and Q3, for which Q3 is the relative depth. So for example, over here, we see the lower part of the heart, and the different colors are the different Q3 coordinates. And when we look at Q1 and Q2, they parameterize the surfaces of constant relative depth, meaning the surfaces of constant color. Now, why do we do this? Well, when we put these heat coordinates into the diffusion tensor, we see that it becomes a block diagonal form, and this will make the calculations in the second step easier. What is this second step? Well, in the second step, we will average out the thickness of the cardiac wall. And for this, we have to define the thickness average of a function f, that is depending on the heat coordinates. And this thickness average is basically the same average we know from statistics, but for which we have to add a Jacobianian, because due to this anisotropic diffusion of the transmembrane potential, the infinities of volume elements will be deformed. For example, we can define the thickness average of the diffusion tensor, which we will denote by HAB. And we can also define the thickness average of the state variables that we use in the reaction diffusion equations, and these we denote by U bar. Now, what is this thickness averaging? For this, we take the three-dimensional reaction diffusion equations, and we start a perturbation scheme in epsilon, which is to be considered very small since we are in a hurricane regime. And when we put this perturbation scheme into the three-dimensional reaction diffusion equations, and we average over the thickness, we get the following result. So when we look at this first equation, we see that the leading order terms are completely determined by the averaged variables, being HAB and U bar. And when we rewrite these equations, we get this end result, for which we again get a reaction diffusion equation depicted in blue, but now in 2 dimensions. So we get a reduction of 1 dimension, and also for which we have to change the derivative by the covariant derivative. And we also get an additional term. Now, what does this mean, this equation, and how can we interpret it? And can we validate this equation? So by looking at this equation, we can say that we can approximate a filament by a spiral wave that moves on a 2-dimensional surface, but this surface is curved in the sense that we had to change this derivative by the covariant derivative, for which the metric is given by the average thickness, by the averaged diffusion tensor. And we also get an additional term, which is some kind of gradient of a potential. And this potential we denote with L, and this is called the effective filament length or the effective wall thickness. And the main reason for this is that in the simplest case, L is given by the traveling time along the thickness of the hard wall. Now, why is this important? When we do some simulations, we can treat the 3D layer as a 2-dimensional surface for which the 3D effects are captured by this potential, and this will go faster. Now, to get a better understanding of this equation, we can project this equation to the degrees of freedom of the spiral wave by using response functions. And afterwards, we can also average over 1 rotation period. And the end result, by doing this, we get this equation, for which n is the unit normal vector to the epicardium, so the outer layer of the heart wall. And what does this equation say? Well, it basically gives us the drift velocity of a rotor in the anisotropic cardiac wall, meaning that if we know the medium parameters gamma 1 and gamma 2 that can be calculated using response functions, then we can predict the drift of the spiral wave. So when gamma 1 is positive, the rotor will drift towards the area for which the potential is minimal. And when it is negative, towards the area for which the potential is maximal. We already did some simulations to validate these equations. for example, in a slab. And we encountered several parameters. So we have different diffusivity ratios, which we see in the columns. We also have different thicknesses of the cardiac wall, seen by the different colors, and also different total fiber angles, which is the total difference that the angles the fibers make, with respect to a fixed axis, which is over here the angle alpha minus the angle the fibers make with respect to the x-axis. And when we look at these simulations, we see that the drift in the gradient direction and the ones perpendicular to it are a good approximated by our theory for small thicknesses, so for smaller than 3 millimeter. We also did some simulations in a human heart geometry, more specifically for the right ventricle. And we treated this right ventricle as a two-dimensional object on which a potential L effective works. So this potential works on the two-dimensional surface. And when we look at the trajectories of the spiral waves, we see that they move towards two regions, which are here depicted by the white dots, and we call these the attractors. Now, what do we still have to do? We still have to do some 3D simulations to compare the three-dimensional reaction diffusion equations with our 2D theory. In conclusion, we have developed a method to find the attractor sites for electrical rotors in the heart based on medical imaging. And when we know the medium parameters, we can predict the drift of the spiral wave on this two-dimensional surface, for which the 3D effects are captured by the potential. And after some validation, we think we should be able to apply this to the atria and to the right ventricle, since they have small thicknesses. I would like to thank, first of all, Karsten and Lendert for giving me this opportunity, but especially my promoter, Professor Dirks, Dr. Pravdin, Professor Panfilov, and Professor Bigtachev for the collaboration.